invasive biopsies have long been used to diagnose, characterize, and monitor different types of cancer. These biopsies are not ideal for many situations, and they're destructive to the human body. However, my research associates at the University of Arizona and I have been working on a new method of monitoring cancer. There's a specific type of cell that can be used to characterize and monitor cancer, but there's been a consistent problem of how to extract this type of cell from whole human blood samples. We found a way using microchip technology to identify and isolate these cells, and we're now able to observe them as an alternative to invasive biopsies. Thank you.
In this project associated with the University of Arizona Center for Applied Nanobioscience and Medicine, I will be researching the behavior of tumor cells in the blood of cancer patients. Research on this groundbreaking technology is crucial and will pave the way to the future of cancer diagnosis and treatment. Current technology of tumor cell detection is accurate and precise, making it a fail-safe way to diagnose a person with cancer. Funding and support of this project and other research being conducted is essential, as mostly everyone is affected by cancer in some way, whether it be in a family member, a friend, or even in themselves. The technology of tumor cell detection will surely be improved and utilized greatly in the future.
The purpose of the project that I'm working on is to help provide faster diagnosis for people with cancer. Cancer cells are actually much larger and more rigid than our normal cells, so they can cause certain body fluids such as pleural fluid, which is the fluid found in your chest, between the chest and chest wall. It can cause these kind of fluids to become much more thick and viscous. So what we're doing is trying to take very tiny glass beads, and by dropping them through this fluid and measuring how long it takes them to fall, we can determine if the fluid is more viscous, and this indicates the presence of cancer cells. Being able to detect the presence of cancer cells through how viscous this fluid may be means we can diagnose cancer patients much faster, which means we can get them care faster and hopefully have a good ending for them. Thank you.
Well, my project focuses on kind of the way, sounds kind of simple, but the way drops dry on different microscope slides. So basically what we're doing is we're taking microscope slides that are coated in various materials, and we're placing drops of various solutions on those slides. And we are imaging them to see kind of what patterns the drops make on the slide. And what we're eventually hoping to do with that is to create like a database of these different patterns, and create so that the database we can use to kind of backtrack with, if we have an unknown sample, then we can drop that on these slides containing coated in the various solutions, and then imaging this unknown sample on these slides, we can kind of put that in the database and try and, like I said, backtrack and figure out what this unknown sample is. And we're hoping to use that to, like with blood, and find biomarkers with, like with cancer patients, different biomarkers on the various cells. So that is basically what the project is focused on, just determining what these unknown samples are through this pattern database sort of thing, I guess, software.